Hi children, good morning all. Last class I have discussed about the structure and gram staining of bacteria, and only 2-3 of you have submitted the homework. What about Punita Shri and Dhanushri? You people never Dhanushri is sending homework then and there. Punita Shri is not sending homework at even a time. Punita Shri, please follow the homework and send it to me. Lavanya, Aishwarya and Vishal are sending their homework regularly. Children, please do the homework properly. And today we are going to start with life process in bacteria. So, based on the respiration, bacteria are classified into two types. One is aerobic respiration, another one is anaerobic respiration. So, what is aerobic respiration? You know very well. It is a basic thing only. The animals or any organism which utilize oxygen for respiration is called aerobic respiration, and those bacteria which are not utilizing oxygen for respiration are called anaerobic respiration. Okay, so let's see about the aerobic respiration. This bacteria require oxygen as a terminal acceptor and will not grow under anaerobic condition, that is in the absence of oxygen. So example for aerobic bacteria is Streptococcus. Next one is obligate aerobes, that is some Micrococcus species or obligate aerobes, that is they must have oxygen to survive. They must have oxygen to survive. In the absence of oxygen, it will die. And this is called what? Obligate aerobes. Next one is anaerobic respiration. You know very well. Bacteria do not use oxygen for growth and metabolism, but obtain their energy from fermentation reaction. It is called anaerobic respiration. Example for anaerobic respiration is Clostridium. Facultative anaerobes. There are bacteria that can grow either using oxygen as a terminal electron acceptor or anaerobically using fermentation reaction to obtain energy. When a facultative anaerobe such as E. coli is present at a site of infection like an abdominal abscesses, it can rapidly consume all available oxygen and change to anaerobic metabolism, producing an anaerobic environment, and thus allow the anaerobic bacteria that are present to grow and cause disease. Example, Escherichia coli and Salmonella. And next one is capnophilic bacteria. What does mean by capnophilic bacteria? Bacteria which require carbon dioxide for their growth are called as capnophilic bacteria. Example, Campylobacter bacteria. Campylobacter bacteria is an example for capnophilic bacteria. That is, carbon dioxide is essential for the growth of bacteria is called capnophilic bacteria. Example is Campylobacter. Next one is nutrition. On the basis of their mode of nutrition, bacteria are classified into two types, namely autotrophs and heterotrophs. Autotrophic bacteria, bacteria which can synthesize their own food, are called autotrophic bacteria. They may be further subdivided as photoautotrophic bacteria and hemoautotrophic bacteria. Okay, two types of autotrophic bacteria are there. So you know very well, either plants or bacteria, if they make their own food, are called what? Autotrophic, isn't it? So among the autotrophic bacteria, first one is photoautotrophic bacteria. From the word itself you could understand that photo means what? Light. By using light, bacteria can prepare their own food. That is, those bacteria are called photoautotrophic bacteria. See, bacteria use sunlight as their source of energy to synthesize food. They may be photolithotrophs. Example, green sulfur bacteria, purple sulfur bacteria, and photoorganotrophs. Example, rhodospirillum. Okay, or the two types of autotrophic bacteria. First one, photolithotrophs. In photolithotrophs, the hydrogen donor is an inorganic substance. For autotrophic, hydrogen donor should be there. Here in photolithotrophs, hydrogen sulfide as hydrogen donor, sorry, in photolithotropes, the hydrogen donor is an inorganic substance such as hydrogen sulfide. Okay, so according to the organic substance, photolithotropes are divided into two types. One is green sulfur bacteria. In this type of bacteria, the hydrogen donor is hydrogen sulfide and possess pigment called bacterioviridin, example chlorobium. So photolithotropes. In photolithotropes, the hydrogen donor is an inorganic substance, you know very well. Based on the inorganic substance used, photolithotropes is divided into two types, green sulfur bacteria and purple sulfur bacteria. In green sulfur bacteria, the hydrogen donor will be a hydrogen sulfide and the pigment present in the bacteria is called bacterioviridin, example chlorobium. Next one is purple sulfur bacteria. For bacteria belong to this group, the hydrogen donor is thiosulfate. Here hydrogen donor will be a thiosulfate and the pigment present in the bacteria is called bacteriochlorophyll. Chlorophyll containing chlorosome are present, example chloromatium. Next one is organotropes. They utilize organic acid or alcohol as hydrogen donor, example purple non-sulfur bacteria rhodospirillum. That's it, children. Next we will see about hemoautotrophic bacteria. They do not have photosynthetic pigment, hence they cannot use sunlight energy. This type of bacteria obtain energy from organic or inorganic substances. So in hemoautotrophic bacteria, they lack pigment. They don't have any pigment in its body. So since they don't have any pigment, they cannot absorb the sunlight for the energy purpose. So what they will do, they will choose as an energy purpose from organic and inorganic substance. Next, hemolithotropes. In this there are many types. Hemolithotropes, this type of bacteria oxidize inorganic compounds to release energy, example sulfur bacteria, thiobacillus thioxidans, iron bacteria, ferrobacillus ferroxidans, hydrogen bacteria, hydrogenomonas, nitrifying bacteria, nitrosomonas and nitrobacter. So these are the various types of bacteria of hemolithotropes. Next, hemoorganotropes. This type of bacteria oxidize organic compound. If a bacteria use organic compound, it is organotropes. If a bacteria use inorganic compounds, it is called lithotropes. Okay. So hemoorganotropes, this type of bacteria oxidize organic compounds to release energy. Example, methane bacteria, methanococcus, acetic acid bacteria, acetobacter, lactic acid bacteria, lactobacillus. Then heterotrophic bacteria, they are parasites. Example, mycobacterium, saprophytes, example bacillus mycoides, or symbiotic, example rhizobium in root nodules of leguminous crops. Reproduction. Okay, this from this we have seen various types of bacteria based on the respiration and based on the nutrition. Based on the respiration, two types of bacteria, one is aerobic, another one is anaerobic. And based on the nutrition, two main types, autotrophic and heterotrophic. Under autotrophic bacteria, two main types, photolithotropes and photoorganotropes. Same way, under hemoautotrophic, there are two types. One is same as lithotropes and hemoorganotropes. Next, reproduction in bacteria. You know very well what is reproduction. Producing young one is called reproduction. Bacteria are reproduced by various methods such as binary fission, gonidia, endospore formation. Among this, binary fission is the most common one. Binary fission, under favorable conditions, the cell divides into two daughter cells. The nuclear material divides first and it is followed by the formation of simple median constriction which finally result in the separation of two cells. Okay, under favorable conditions, the cell divides into two daughter cells, right? The nuclear material, first nuclear material divide into two and is followed by the formation of a simple median constriction in the cytoplasm and separation of two cells. By the diagram itself you could understand, children. Next, endospore. You know this endospore formation. During unfavorable condition, bacteria produce endospore. Endospores are produced in Bacillus megaterium, Bacillus sphaericus, and Clostridium tetani. What is endospore? Endospores are thick-walled resting spores. During favorable condition, they germinate and form a bacteria. So during unfavorable condition, the bacteria secrete thick wall around it and become a spore. This spore resisted to the unfavorable condition. During favorable condition, the spore germinate and become a bacteria. This is called what? Endospore. Next, sexual reproduction. A typical sexual reproduction involving the formation and fusion of gametes is absent in bacteria. However, gene recombination can occur in bacteria by three different methods. They are called conjugation, transformation, and transduction. So you know very well, in a typical sexual reproduction, fusion of male and female gametes has taken place, isn't it? In bacteria, bacterial sexual reproduction, such kind of fusion will not be taken place, but there is a gene combination, that is gene transfer from one bacteria to another bacteria and combine in that bacteria, so that we can consider as a sexual reproduction. There are three methods of gene recombination. One is conjugation, transformation, and transduction. What is conjugation? Liederbeck and Edward L. Tatum demonstrated conjugation of E. coli in the year 1946. In this method of gene transfer, the donor cell gets attached to the recipient cell with the help of pili. The pillars grow in size and form the conjugation tube. Then the plasmid of donor cell, which has the F plus, that is fertility factor, undergoes replication. Only one of the strand of DNA is transferred to the recipient cell through the conjugation tube, and the recipient complete the structure of double-stranded DNA by synthesizing the strand that complement the strand acquired from the donor. Okay, what they are trying to say in conjugation tube, see, in each and every bacteria there is a extra chromosomal ring called plasmid is present in the lateral side, isn't it? This plasmid one is a double-stranded DNA. This double-stranded, among these two strands of DNA, one of the strand of DNA is passed through the conjugation pilus from a donor cell to recipient cell. In the recipient cell, the single strand of DNA synthesize the another strand of DNA using the complementary DNA. This is called what? Conjugation. Next one is transformation. Transfer of DNA from one bacterium to another is called transformation. So when a DNA of one bacteria is transferred from transferred to another bacteria is called what? Transformation. In 1928, the bacteriologist Frederick Griffith demonstrated transformation in mice using Diplococcus pneumoniae. Two strains of this bacterium are present. Okay, children, this is what they are trying to say. With the help of diagram, you can identify. There are mice. Mice, when there are two strains of bacteria are there in pneumococcus. One is R strain, another one is S strain bacteria. When bacteria R strain is injected into the mouse, it will be alive. When S strain bacteria injected into the mice, the mice will be dead. And the same time, heat killed S strain will be injected into the mice. What happened? The mouse is live in condition. But when we inject the mixing of R and S strain, heat killed S strain into the mouse body, what happened? The mouse started to die. Why? Because the R strain change the estine into a virulent form and become a toxic and change into what? Death, cause a death. This is what? Transformation. Next one is transduction. With the help of diagram, please you understand this concept, children. I am going fast only, I know very well, but no other go. Next one is transduction. Zinder and Liederbeck discovered transduction in Salmonella typhimurium. Page mediated DNA transfer is called transduction. Page mediated DNA transfer is nothing but the genetic material of one bacteria is transferred by a virus is called what? Page mediated DNA transfer. When a bacteriophage transfer the DNA genetic material from one bacteria to another bacteria is called transduction. There are two types of transduction: generalized transduction and specialized or restricted transduction. In generalized transduction, the ability of a bacteriophage to carry genetic material of any region of bacterial DNA is called generalized transduction. Okay, that is carry or bacteriophage carry genetic material from any region of bacterial cell is called what? Generalized transduction. What is mean by then specialized or restricted transduction? The ability of the bacteriophage to carry only a specific region of bacterial DNA is called what? Specialized or restricted transduction. So these are the two types of transduction. So transduction is nothing but transformation of genetic material from one bacterial cell to another bacterial cell by bacteriophage is called transduction. There are two types of transduction: generalized transduction and specialized transduction. What is generalized transduction? In generalized transduction, the bacteriophage can have the ability to transfer the DNA from any region of its body is called generalized transduction. What is specialized transduction? It is nothing but the bacteriophage can carry only a specific region of the bacterial DNA is called specialized or restricted transduction. Next topic, economic importance of bacteria. Please go through by yourself, children, this tablet column. Then plant disease caused by bacteria, animal disease caused by bacteria, human disease caused by bacteria. Also please do yourself, children. Next I am going to see about archaebacteria. Archaebacteria are primitive prokaryotes and are adapted to thrive in extreme environment like hot springs, high salinity, low pH and so on. So what is archaebacteria? It is nothing but the bacteria which can survive in an extreme environment, okay, such as acidic medium or hot environment, like that. They are mostly hemoautotrophs. So archaebacteria are mostly hemoautotrophs. The unique feature of this group is the presence of lipid like glycerol isopropyl ethers in their cell membrane. Their cell membrane possesses unique features such as glycerol and isopropyl ethers in their cell membrane. Due to the unique chemical composition, the cell membrane show resistance against cell wall antibiotic and lytic agent. Example, Methanobacterium, Halobacterium, Thermoplasma. Next one is Cyanobacteria. Cyanobacteria, otherwise called as blue-green algae. How old are Cyanobacteria? Stromatolites reveals the truth. Stromatolites are deposits formed when colonies of cyanobacteria bind with calcium carbonate. What is stromatolites? Stromatolites nothing but the colonies of cyanobacteria bind with calcium carbonate and form some rock-like structure, and this is called what? Stromatolites. When we take the life of the stromatolites, it reveals the life of the cyanobacteria. Okay, they have a geological age of 2.7 billion years. Okay, 2.7 billion years ago itself cyanobacteria are evaluated on the earth. Their abundance in the fossil records indicate that cyanobacteria helped in raising the level of free oxygen in the atmosphere. Very good. And cyanobacteria are popularly called as blue-green algae or cyanophyceae. They are photosynthetic prokaryotic organisms. According to evolutionary records, cyanobacteria are primitive forms and are found in different habitats. Most of them are freshwater and few are marine. Example, Trichodesmium and Dermacarpa. Trichodesmium erythraeum, a cyanobacterium, imparts a red color to Red Sea. Please mark it. Then when the Trichodesmium erythraeum present in the sea, it gives a red color to the seas. Then species of Nostoc Anabina lead an endophytic life in the coralloid root of cycas leaves of aquatic fern. Azulae by establishing a symbiotic association and a fixed atmospheric nitrogen. Members like Glucopas caspa and Nostoc Cytonema are found as pycobionts in lichen thalli. Salient features. The members of this group are, it is a 5-mark question, children. Prokaryotes and lack motile reproductive structure. Thallus is unicellular in Crococcus, colonial in Cloacopsa, and filamentous in Trichome in Nostoc. The gliding movement is noticed in some species. The protoplasm is differentiated into central region called centroplasm, peripheral region bearing chromatophores called chromoplasm. The photosynthetic pigments included C-picoxanthine and C-picoerythrin along with myxoxanthine and myxoxanthophyll. Reserve food material, xenobicin starch. In some forms, large colorless cell is found in the terminal or intercalary portion called heterocyst. Then they reproduce only through vegetative method only. The presence of mucilage around thallus is characteristic feature of this group. Therefore, this group is called Myxophyceae. Then sexual reproduction absent. Microcystis, aeruginosa, Anabina flows aquae, cause water blooms and release toxin and affect the aquatic organism. Most of them fix atmospheric nitrogen and used as biofertilizer. Example, Nostoc Anabina. Spirulina is rich in protein, hence it is used in single cell protein. Thallus organization and methods of reproduction is given in the figure. Okay, children. Next, mycoplasma or molecules. I will start in my next class, children. And the homework is the types of bacteria based on the respiration and nutrition. Or the 2 questions, please complete it. Bye. I will get you in my next class.